I kindly request our organizing secretary, Dr. S. Jagadishri ma'am, to kindly honor our today's judge for paper presentation, Dr. J. Mukesh Kumar from Bharat University and Dr. Balakumran sir from Dwarkadas Govardhandas Vaishnav College. I welcome you, ma'am. Thank you so much, ma'am. Now our paper presentation is about to begin. The first presenter will be Satish Kumar from DG Vaishnav College.
A very warm welcome to everyone present here. Myself, Satish Kumar from Microbiology Department of Dwarkadas Govardhandas Vaishnav College. I would like to present an hypothesis on starch clay polymer, a novel food packaging material, under the guidance of Dr. P. Vidya, head of our department. Next slide. These are the contents that I will be dealing with. First comes the introduction. As we all knew, there are many kinds of food packaging materials available in the market at present. These food packaging materials are not only hazardous to the environment but also to us. These food packaging materials, when manufacturing these food packaging materials, they produce various greenhouse gases. These greenhouse gases pollute our environment that eventually affects our body, and we get various kinds of health problems like cancer, obesity, and cardiovascular disease. They may be short or long term. Next slide, please. According to an health scientist, an average person needs about 1.4 kilograms of food per day. We actually produce that implies that we will be needing about 3.7 billion metric tons of food per year. But we actually produce 4 billion metric tons. Then why does this kind of error as hunger? Because we are not only producing food, but we are also wasting about 1.7 million tons of food per year. This is a chart that represents the progression of food waste that has been produced according to the regions by the World Bank. This chart, this blue bar represents the year 2016, and the yellow represents for 2030, and the red for 2050. Next slide, please. Next is objectives. My main objective is to find an ideal material that is suitable to produce this kind of food packaging material. Personally, I think that this ideal material is starch and clay. Then I need to construct the material that prolongs the shelf life. Because we are producing a food packaging material, I need to prolong the life of food by any means possible. Next, we are not only producing a food packaging material, but also we are producing an eco-friendly food packaging material. So I asked to prove the biodegradable nature of it. Next slide, please. Next is the methodology. This is the methodology I'm performing now. Actually, this is one of my projects, MSc project that I've been pursuing. So I'm just pursuing the hypothesis of it. My method is to first collect and starch from a starchy food waste that is available in the country. Next, I have to synthesize some clay nanoparticles because nanoparticles are what to say. Nanoparticles are said to be more effective than normal particles. Next, I need to synthesize clay and starch into a biopolymer composite by melt processing. Next is the drying process. Next slide, please. Each and every product that is in the market has to prove his worth by passing and physical and chemical properties of characterizations. So the physical and chemical characterization of this material plays a vital role in the methodology. Next, packaging material formulation. A material cannot solely survive by its means to protection, but also by the type of its appearance. A packaging material must be attractive so that it is easily available and easily what to say, it is easily attracting the customers. Next, the food preservation testing. As we are producing a food packaging material, it has to preserve our things. So it should pass the food preservation testing. Next comes the biodegradable nature. Its biodegradable nature must be what to say, must be effective so that it is said to be a biodegradable eco-friendly polymer. Next slide, please. Next is the hypothesis. I commonly call this 5 whys. First why is why starch. Starch, as we all know, it is a mostly abundant material in the nature. And thesis states that a starch that is actually about 37% of starch in an average food waste. So why we should not use this starch and go for a not how to say difficultly acquired petroleum derivatives. So we can use this easily available starch as an alternative easily. Next, why clay? Clay is not yeah, you may say that clay is a soil that is not easily available in our country, but this clay is from the ancient times. This clay plays a major role in food preservation, not only as a food preservation but also as a it has many healing capabilities. Next, why starch and clay? Each and every person has their own defects. Likewise, starch and clay has their own defects, but these defects, when come together, is getting neutralized. This I made a review, so it came that this equally what to say, they are neutralizing their own defects. So next, why the food packaging? Yeah, as I said that this clay has many healing capabilities. Then why should I use for food packaging? Clay is said to have an entrapping capability that entraps ethylene oxide, that is ethylene gases, that is usually responsible for fruition of fruit or vegetables. So this clay traps the ethylene gases, which delays the fruition of the vegetables. So it extends the preservation time. Won't clay soil be exploited? You may say, yeah, by using it rapidly, then we will be exploiting it. But if it is biodegradable in a rapid state, we can use it multiple times in a frequent period of time. Next is my conclusion. End is here, but not for us, but for the problems that needs a solution. Thank you.
Any questions?
Good afternoon.
Good afternoon, sir.
Actually, are you going to work it out or it's already done?
It's already in work, sir.
Okay.
So I've been processing certain materials, so it's on the process.
So why you didn't display any experimental images in this presentation
No, sir, it is not officially been published, so I'm saying it.
officially? Okay. So in your five whys, no, why?
Yes, sir.
Actually, in the methodology, you have mentioned like you are going to synthesize clay nanoparticles,
Yes, sir.
but nothing you have mentioned in that the why questions. So
Okay, about
how
the
you are
nanoparticle.
relating the starch, clay, and the nano?
So actually, what I was processing was I'm going to process clay into nanoparticles
Okay.
that will be entered with essential oils. So these essential oils prevent more seeping of water. So the seeping, if the seeping of water is being stopped at a particular time, the starch won't degrade in the rapid state unless until any microbes is in presence.
To make a composite, are you using any binders or
No, sir. Actually, I have not kind of binders. As a plasticizing agent, I'm using glycerols and gelatins, glycerol and water.
okay? Thank you.
Thank you, sir. Sorry, sir.
Yeah. Satish, you are mentioning something about nanoclay, right? It's
Yes, sir.
basically about the silica based one. It's
Yes,
not-
sir.
Silica, I feel it's a bit difficult to control the size and other stuff.
Okay.
So how are you going to do that?
Um,
Or you tell me how you synthesize the clay nanoparticle in general.
in
You didn't come up to
yes, sir.
how will you synthesize?
Um,
What is the what the synthesis process?
so according to the paper I reviewed, that majorly they are doing this synthesizing nanoparticles by physical processes only, sir. So they are been crushing for into particular sizes, sir. Because those are like what to say. The silica can be sometimes can be a bit of
Mic mic
yes, sir.
What is the charge of your charge as well as your clay? I'm asking what is the perceived charge of your clay?
Charge?
Because any particle is
Yes,
not
sir.
a charge, right?
Yes, sir.
No particle charge is a charge.
Yes.
What about your clay?
Clay is actually basic, sir, so it will be a positive charge.
Sure. When charge,
Starch is kind of positive, but when heated,
last
how
time
is going to get a nanoformable material?
that's why I'm using plasticizing agents, sir. So these glycerol and water it makes it easily to combine, sir.
It will be a binder. The green thing.
Yeah, yes, sir.
Property?
Um, property, according to the paper I reviewed, it is actually stable, pretty stable, sir.
Sure.
Yes, sir. Thank you.
Our next presenter will be M. Nisha from Valyamal College.
பண்ணிருப்பேன்
So, good afternoon, everyone. So, my name is Nisha. I'm studying second MSc Biotechnology, and I'm from Malimal College for Women. So today I'm going to present a review on the role of gut microbiome in regulating human physiology and pathology. So before we move on to the topic, I want to talk about microbiomes. So microbiomes represents a group or a community of microorganisms that resides in a particular area or a region. So microbiome refers to a group of microorganisms that lives in a particular area or a region. In my case, it's going to be the human gut. Next slide. Next slide. So now that we know about microbiomes, let's talk about gut microbiomes. So the microbial ecosystem that is present in the gut of any living organism is commonly referred to as gut microbiomes. The gut microbiome is very unique to each and every individual, and it can be influenced by various of factors like diet, antibiotics, and environmental exposures like stress, the depression, anxiety, sleeping habits, etc. So now we are going to talk about the composition of gut microbiome. The gut microbiome is made up of various of microorganisms like bacteria, virus, fungi, etc. But the most predominantly found microorganism belongs to these species: Bericomicrobia, Firmicutes, Bacteroidetes, Proteobacteria, and Actinobacteria. When it comes to fungi, we have Aspergillus and Candida as the predominantly found microorganisms, and for virus we have the viral component of the gut microbiome is called as viriome, and we also have bacteriophages called CR asphage. So this is the main part of my presentation, the functions of human gut microbiome. So when I was reading about human gut microbiome, I realized that I was reading about an invisible world that was living inside me. So, human gut microbiomes possess various functions in our body, and it is fascinating to think that these teeny tiny microorganism is capable of impacting a whole physiology. Next slide. So first and foremost function we have here is digestion and metabolism. Since they live in the gut, the first and foremost function they do is the digestion and metabolism of the food that we eat. The microorganisms present in a gut microbiome will break down the indigestible foods that we eat into complex carbohydrates and fibers, and also helps in absorbing the nutrients from them. And they also regulate glucose and insulin levels by producing glucose regulating hormones, and also by synthesizing short chain fatty acids by converting the dietary fibers into SCFAs. The SCFAs in turn will influence the glucose metabolism by increasing the insulin sensitivity. Then we have immune regulation. We all know that gut is one of the physical barriers of our immune system. So the gut microbiome is also a part of our immune system. The gut microbiome supports the immune system by producing cytokines and also by regulating them to fight the infections. The various studies have been shown that there is a relation between the gut microbiome and the immune system. Researchers have done studies on germ-free mice to prove that the germ-free mice that is the mice which lacks the gut microbiota, that it has low immune response when compared to a normal healthy mice. Then we have fecal microbiota transplantation procedure, which has proven to help strengthen one's immune response. And also we have comparative study of gut microbiomes between a healthy person and a diseased person. And lastly, we also have in vitro studies where researchers have proved that the gut microbiome is indeed capable of producing immune regulating hormones. Then the very important function of a gut microbiome, it is the mental health. The gut microbiome is said to have been linked with a gut-brain axis, and it also regulates hormones and also produces neurotransmitters. The gut-brain axis is influenced by the gut microbiome by producing neurotransmitters like serotonin, dopamine, etc., and by also modulating the HPA axis, that is the hypothalamus, pituitary, and adrenal axis, which has been responsible for the stress response. The studies have shown that the imbalances in the gut microbiome have been the imbalances in the gut microbiome have been linked to mood disorders like depression and anxiety. So whenever a person has a depression or an anxiety, they can be said to have low gut microbiota, or they have an imbalanced gut microbiota symptom called dyssymbiosis.
Second.
Again, there have been studies showing that the gut microbiota is related to mental health. For example, we have animal studies in which the scientists have changed the gut microbiome through antibiotics or through the diet, and it has resulted in the behavioral and mood change of the animal. Then we have cardiovascular health, in which the cardiovascular health was influenced by the cholesterol metabolism and the blood pressure regulation by gut microbiota, and metabolic pathway and studying metabolic pathways, inflammation, lipid metabolism, and blood pressure regulation has proved the function of gut microbiota in cardiovascular health. And we also have vitamin synthesis, in which vitamin K and vitamin B are synthesized by gut microbiota, and again various studies have been done to prove this function. Then recently we have discovered the functions like the gut microbiota relating the gut microbiota with neurological disorders, inflammatory bowel disease, and obesity and weight management. So this is the characterization analysis method we use for the gut microbiota. And then we have conclusion. That's it. Thank you.
Okay, in the title itself, you have said it's a review. Okay, so my suggestion. So whenever you are presenting a review one, okay, kindly include the references.
So last slide.
Last slide coming up.
Conclusion.
Okay, so if possible, you can put it in the content also. And whenever you adopted so by my observation, okay, the presentation was nice, but it should be more in the only word.
Specifically mentioned about microbiota.
Genus cap, full caps. Capital
Recently, a lot of information was portrayed about the mutation happening in the microbiota. So,
The gut microbiota has been, as the days go by, we are converting a natural diet to more into a fast food diet. So we are also our sleeping patterns are also going to be irregular. So when these things happen, our gut microbiota is also going to be influenced by these environmental factors. So the good gut bacteria is going to be more is going to be evaded by the bad bacteria, and the mutation is going to occur.
that is fine. After mutation, basically the pathway is not going to be reduced. The output pattern is not going to be changed.
Yes, sir. So Helicobacter pylori was a good bacteria. First when we had first when it was a part of a gut.
Solution, I'm asking. Is
Yes.
never going to solve are you going to include some other new features or something like that so that can regulate the microbiota and things like that? Do you have any kind of review of it on your?
Only we have fecal microbiota transplantation, sir, right now.
பார்த்தேன் மேம் இப்போ பண்ணிட்டு இருக்கேன் மேம்.
Our next presenter will be Sairam G from DG Vaishnav College.
Good evening, sir, and everyone. This whole presentation is based on this one single cut. Just take a 1 second and look at this. Said by Charles Darwin. So this presentation based on current state of artificial intelligence, challenges and opportunities. And myself, Sairam, from Department of Microbiology, DG Vaishnav College. I am presenting this paper under the guidance of Dr. M. Abhirami ma'am. So these are the table of contents. So I am just I don't give any brief introduction about AI. I am just give a glimpse of AI. What is AI? AI is nothing but a technology or an ability which we are applying to the mission just to mimic a human, all the senses of human and their ability. Even in some extent, AI can perform which we can't do in a single day. When a research scientist is looking for some article, he can refer about maximum of 9 articles as per Times of India recorded. But with the help of AI, you can analyze millions of articles within a hour. AI is nothing complex. It's like a bioinformatic tool which we can introduce into life science, but just by giving all the biological data from NCBI, ICMR, and US Food and Drug Administration, and we can fine tune based upon the domains we want, and even we can't also able to predict what will be the result. This can easily cut off the time, and I may not show, but it can have the possible to eradicate the wet lab practice in some extent. So these are the hierarchy of AI. Also, sorry, heredity of AI. First one is reactive missions, which was initially came in 2010. This one is you can see in every mobile phone which have face recognition. This is the module that we have in every face recognition module. This relative mission principle is behind that, which can sense that external stimulus and your operations. And many have imposed to Snapchat that filters how can it perfectly fitted into your face. This based on this relative mission. Next one is limited memory. In limited memory plays a huge part in librarical or journal sessions, which we can feed some data. It can fine tune themselves. Even if you give some updated data, it will surely fine tune themselves. We don't have to set some manual method or someone who have to code every time. And then theory of mind. Theory of mind is a third stage of AI, which has the sense and response to the human nature, what our emotions is, how we are expecting. Even some AI bots are based on this. In Delhi also, a college student worked on this project called a psychological bot, which you can instead of visiting therapy centers, you can talk with them. They usually react with they usually carry the same emotions which you want. And the last one is self-aware. Self-aware is the advanced AI technology, which you can many of them known that ChatGPT and Adobe AI, Wonder AI, many of them which has introduced in 2023. This all comes under third generation of GPT transformer principle, that is self-aware. Now in life science, AI has the potential to revolutionize the entire life science by minimizing that time consumption which we take to produce results, because it has the ability to predict with what data we have given, and then it usually in major in current trends, in major application they are focusing on drug discovery. They usually used to analyze and like in bioinformatics they used to dock proteins, and they can predict some data which we can in bioinformatics in some extent some cannot be visualized, but in AI it can be visualized in 3D perspective, and in some it can be as a live prototypes. uh, these are the major domains of application currently AI is using. In first one, drug discovery lifetime. Likewise, I say it's similar to bioinformatics. They use genomics and proteomics data just to discover some novel drug. If we can assume that this drug is novel by referring certain set of data, but with AI we can check whether it is a novel or already existed within a few seconds. And in precision and in medicine is used to analyze a patient's disease characteristic, and what are the treatments they required. And in disease diagnosis like X-ray, MRI, and PET scans to give more accuracy of the results. So these are the notable outbreak researches performed with AI. IBM is the technological company everyone knows. They introduced Watson, a cognitive computing. Cognitive is nothing but how to say, it's like a reaction process. Understanding process. They used this cognitive. Initially, Watson was discovered for the business analytics. Later, they included in the life science to promote drug discovery and their targets. And that one is Halcyon compound. Halcyon was initially stopped. Okay. So Halcyon is a compound which is previously used in diabetic drug. Later it discontinued for its weak efficacy. But everyone, we are battling 2022 in COVID. MIT US have identified that Halcyon is the only compound which battled almost 600 molecules which is responsible for the antimicrobial resistance, and they are sorry, and they also evident that it is the first drug that the first molecule that was identified with the help of machine learning. If the machine learning is not there, it is surely huge loss for many of us. And my conclusion is it will be surely see in some extent it will be a cause a threat to every life science students, because their competency of knowledge is somewhat high.
First one மட்டும்
We are in we are in extremely base stage when compared to even I'm not generalizing, but I'm sure that many of them have not exposed till what is AI, and even I don't know what is detailed about bioinformatics. So try to fit with technology what are all emerging, so it will be sure shot to find new novelties. Thank you.
ஃபஸ்ட்டு மட்டும் இதே பிபிடியை வேற போய் வாசிக்கணும்.
It's a nice presentation.
Thank you, sir.
Actually, even a few months before, I have shared in my WhatsApp status, okay, especially those who are life science students. Okay. So we must push ourselves into AI, IoT, machine learning, all those things because
இல்லை இப்போ
when compared with other CS people,
ஒன்றும் இல்லை
okay, we are very lack.
அதே ஃபைல சீவாசன் கொடுத்தா 2 வாரம் செய்யும்
That's my
உள்ள
opinion.
தனியா வராது
Okay,
delete
so
பண்ணாம save பண்ண முடியும்
பிபிடியாக சேவ்
even we have to learn
delete
many
பண்ணிட்டு எல்லாத்தையும்
things like Python. Okay, so all those things, it's not only for CS or other IT people. Okay, even life science, okay, can get
அவ்வளோதான் வேறு வேறு
this kind of knowledge, and
எடுத்துக்கிறேன்
we have to be ready for the next generation of opportunity. If we still in the laboratory and working out with the experimental part, you know, we will be very lack. Okay, and especially that Chat GPT recently, it's, you know, I don't know what to say.
Sir, one second. Sir, it's usually like now it's like a puppy type. They are training with more knowledge. What actually we are the one, we are inputting some knowledge what is the time of USA current right now. Without you can even Google is an hybrid search engine, but we use when we use ChatGPT, it gives the accuracy and
Yes, yes.
yes.
Anyway, this is the future of everything. Thank you.
Sir, I want to mention one thing. So, likewise, Sir said just to explore what is it. We don't want vast knowledge about this. Just to four basic knowledge in these four domains, like Python. What is Python? What is data science? What is data analytics? And what is CSS? This will show shortly take us into next level for a new novel drug discovery, which results in which we can cure many diseases and which we can prevent any COVID like what we suffered earlier. Thank you.
மேம் அனுப்பிட்டேன் மேம்
பேக் ஆச்சு
அவங்க நம்பர் இல்லையா மேம் சரி சரி ஓகே மேம்
ஷுடா நான் பண்ணேன்
Our next presenter will be Vishnupriya and Lokeshwari from Dhanapalan College. Good evening to one and all present here. I'm V. Lokeshwari. This is C. Vishnupriya from Professor Dhanapalan College of Science and Management. I'm hereby to present a paper in the topic of applications of ozone-treated gingiber officinal extract for the biopreservation of blueberry. Next slide, please. Okay. First of all, why we want to preserve the food? Because it leads to the contaminations of food. That leads to the contaminations of food. The microbes release some of the toxic substances in this food or any food products. The toxic substances is ingested by the humans, which leads to some of the diseases and disorders. So we need to preserve the food. So we here, instead of chemical additives, we use a biopreservation techniques by using a ginger extract, gingiber officinale extract. Next slide. So we take blueberry. Blueberry is a seasonal fruit and it is also rich and expensive, and it is rich in vitamin K, and it plays an important role in the promoting heart health. And this vitamin is also important for both bone health and blood clottings also. So we take a blueberry as a experimental use. Next slide please. So these articles, the aim is to preserve the blueberry throughout the years by using a ozone treatment technique to increase the shelf life of the blueberry by using a ginger extract. Next slide.
Next we go into methodology. We take ginger beer officinale extract like ginger 150 gram. Then we rinse it the ginger and peel out the outer layer. Then we use to mixture to get the pure extract without adding water. The pure extract 10 ml in the sterile beaker, and we take a different ratio in the 5% and 10% extract in the pure extract. First one is 5 extract, 0.5 pure extract, and we add to 9.5 ml water. Then we 10% extract to add with the 9 ml sterile water. Next slide. Then we get to the ozone treated the extract 10 ml to each different
So steps. Take 3 test tubes. So we take a set of 6 test tubes and they contain a different ratio of pure extract like ginger extract 5% and also 10%, and separately 2 test tubes where it has been to be ozone treated. So the 6 blueberry has been dipped into the blueberry into a ginger extract. Next slide. So we ozone treated a 10 ml pure extract with the 50 pp 15 minutes and 10 ml pure extract with the blueberry for 30 minutes. Next slide please. Next slide. Next slide. So the results show that the blueberry in the control spoils within 48 hours because with the controls will dipped into a sterile water only. So 5% and 10% of extract will dipped blueberry has shelf life is increased for 3 days, and the pure extract dipped blueberry so increased the shelf life for 7 days, and ozone treated only increased the shelf life for 10 days as the ozone treated inhibit the growth of the microorganisms in the blueberry contaminations. Next slide please. Next slide. Next. So as I concluded that the bioactive compound present in the ginger like gingerbread which combinations of ozone treated inhibit the microorganism growth in the contaminations of blueberry. So throughout this biopreservation technique we can preserve the blueberry throughout the year. Thank you.
Vishnupriya, right? Can you give me some more highlight how you did this ozone treatment to the gingerbread on practical basis?
So we take test tubes and pure extract of a ginger is taken for 10 ml and different concentration like 5% and 10%, sir. So only set of 3 test tubes only contain pure extract, that is 10 ml only. So we have ozonated for one test tube for 15 minutes and another is 30 minutes.
How did you ozonate it?
So that ozone mission is there, sir.
Show me.
Slide. Next. Next slide. Next slide. Next slide. This one, sir.
Okay. What is the initial pH before ozonation?
Initial pH is 5, sir. I think so. 5.
Okay. Did ozonation have any effect on the pH?
No, sir.
Sure.
Sure, sir. We can also taste the food, sir. There is no changes in the taste also.
See, being ginger is already an antibacterial and biopreservative.
Yes, sir.
Why you want to do an ozonation?
We want to preserve for external use only. So for ginger extract is not only for a blueberry preservation, sir. So for ozonated only, if only preserve the food, sir.
My point is, your ginger extract is already an antibacterial activity.
Yes, sir.
So it can be used as a biopreservating agent.
Yes, sir.
Then why you are enhanced? Did you find any enhancement in the basis of antibacterial activity?
No, sir. No. We there is no changes only.
Then why you need to put an ozonation? Then everything is fine. Why you need to add some more?
Because already a senior only a senior project is ozonated marine seafoods like fish, prawn. So if fruit they have to biopreserve it. So that's why only we use this fruit ginger extract. They use biotic components like turmeric and that's all.
Maybe you would have provided a chart where before and after. So in that case, by doing an ozonation, I can able to find out that the activity was somewhat higher when compared with the one which does not with ozonation.
Oh.
Maybe that could give some more highlight on what exactly we are expecting.
Okay, okay, sir. Okay, thank you, sir.
See, actually, the principle behind ozonation and uh, that's not very clear the way you are sort of presenting. That's the thing sir is also asking. Okay, normally ginger extract, it's no on its own it has antibacterial antibacterial activity.
Yes, sir.
So you must be very clear, okay, by adding ozonated, okay, how it improves the shelf life of. And blueberry, normally recent days we are getting blueberries from supermarkets, okay, they are all from Peru, okay, already almost from USA's, okay, they have frozen containers, everything, okay. So after, here there is no mean of keeping the fruits for preservation. We have only in two forms. One is like a dried one, frozen one.
Yes, sir.
Okay, so for my suggestion, okay, better you can select some other fruits, watery fruits, okay, which is more viable for us.
Okay, okay, sir.
Thank you.
Okay, thank you so much. Good evening, everyone. It gives me immense pleasure to introduce our judges for today's poster presentation, Dr. S. Kanimuri and Dr. K. Krishna Prema. Dr. S. Kanimuri is the head of the department in the Department of Microbiology in Assam Memorial College of Arts and Science. She has appointed as Dean of Academics. She has published several research papers in peer-reviewed journals and ma'am has also attended many national and international conferences. May I kindly request head of the department, Dr. P. Vidya, to kindly honor ma'am with a bouquet. With a bouquet and memento as a token of love and appreciation. Dr. K. Krishna Prema is the head of the department in Department of Microbiology in Ethiraj College for Women. She had received many funds from Ethiraj College Trust for two different projects. She has published several research papers in peer-reviewed journals. Now, I kindly request head of the department, Dr. P. Vidya, to kindly honor ma'am with a bouquet and memento. Both of them are closely associated with the department and a very close friend of our HOD, Dr. P. Vidya, and are seniors of our professor, Dr. S. Jagadishwari. On behalf of PG and Research Department of Microbiology, I heartily welcome both our judges. Welcome, ma'am, and thank you, ma'am.
Our next presenter will be Jeevita M. from Quality Millet.
இருக்கு அங்கே இருக்கு.
Good evening to one and all present here. This is Jeevita from Kaidamalat College. I am here to present my work Biomonitoring of Air Pollution using Nerium oleander and Calotropis gigantea in 2 different pollution zones of India. Introduction. As we all know, air is more essential to live, not only for humans, it's also for plants, animals, etc. But nowadays, air is heavily polluted by our activities. If any changes in their concentration and compositions, it may worsening air quality as well as cause human health concern as well as ecological problems. The main factors of air pollution is rapid industrialization and urbanizations. So now we are forced to figure out how to clean the air with cheap and easiest method. I have mentioned that air purification is most important one, but in my work I just monitoring air pollution by using some plants. Some plant act as sink to absorb pollutant from the air and as well as purify the quality. For my work, I have chosen these 2 plants for my work. First one is Nerium oleander. The second one is Calotropis gigantea. Next. Nerium oleander is commonly known as oleander or Nerium. It's a small shrub as well as small tree which is cultivated throughout the world. It is belong to the family Apocynaceae. As well as Calotropis gigantea belong to the family Apocynaceae. It is a large shrub growing to 4 meters. Next. The main goal of my work was to collect plant samples at 2 different areas. We have heavy polluted area and low polluted area to study total chlorophyll content and pH of leaf extract of the plant, to estimate ascorbic acid content and relative water content in the leaves of the plant samples, to study the APTI, which is known as air pollution tolerance index of 2 plant samples of each 2 places. Next one. Materials and methods. Collection of fresh leaves from these 2 plants at 2 different areas at the early morning. The collected leaves were washed with tap water and rinsed with distilled water and then used for further laboratory studies. This studies is used to predict, assess the air pollutant on the plant surface. Total chlorophyll was estimated by Arnold method. Next. Ascorbic acid content was estimated by Bajaj and Carr method. Relative water content was estimated by Bars and Weatherly method, as well as pH of the leaf samples were estimated by SK Singh method. Next one. Air pollution tolerance index was estimated by Singh and Rao. Next. Results. The concentration of total chlorophyll was decreased in both the plants were collected at high polluted area when compared to low ones. Decreased chlorophyll content indicates that it is an best indicator of sulfur dioxide. Concentration of ascorbic acid. Ascorbic acid content was increased in both the plant were collected at heavy polluted zones when compared to low ones. This is because ascorbic acid is an antisorbic vitamin reducing agent which is play an important role in sulfur dioxide reduction. Next one. pH of the plant. The 2 plants collected from heavy polluted zone exhibit pH change towards acidic range. This is because of the presence of nitrogen oxide of nitrogen and sulfur dioxide in the air, which cause change pH leaves up towards acidic range. It is upon diffusion. It is upon diffusion of SO2 through stomata. Next one. Relative water content. Plant grown from this polluted site shows reduced relative water content when compared to low polluted ones. As we know, relative water content is maintained physiological status under stress condition, including air pollution. Next one. Concentration of APTI. APTI is also known air pollution tolerance index. The calculated APTI was decreased progressively from low to control. The level of air pollution tolerance index, it depends upon the level of total chlorophyll, ascorbic acid, pH of the leaf extract, as well as relative water content. This is the best evidence that the plant grown alongside by roadways exhibit significant reduction in chlorophyll, pH of the leaf extract, as well as relative water content. Next one. Conclusion. Finally, I have concluded the study reveals stress levels created by the vehicular pollutions were higher in the heavy polluted area when compared to the low ones. Both the plants as considered as the intermediate species. Among the two selected plants, Caradox gigantea were found to be most suitable species to be used in the vegetation traffic barriers based on the air tolerance pollutions. Furthermore, air pollution tolerance index determinations provide a reliable method of screening large number of plants with respect to the response to air pollution. Thank you.
Nice presentation. You're a scholar,
Thank you,
right?
sir.
You're
Yes,
a
sir.
research scholar. You're
Yes, sir.
doing your PhD.
Yes, sir.
Which year?
I completed third year, sir.
Third.
This is my fourth.
So, what is the main purpose of selecting these two plants?
Because this has more pharmaceutical characters, sir, as well as this acts as a good bio monitoring plant. Many of people doesn't know about these plants.
Okay.
That's why I choose it. As well as it has cure some air pollution borne diseases like pneumonia, asthma, etc. Sir. That's why I choose this.
Okay. And even in the presentation, you could have included some images.
Yes, sir.
And all the tables, it shows just numerics, but it's not like statistical oriented results. Okay. Because see, you're a research scholar.
Yes, sir.
Okay, so in your slide it should be like a research scholar.
Okay, sir.
Okay. Thank you.
Thank you so much, sir.
Kindly maintain silence. Our next presenter will be Jemima from DG Vaishnav College.
Good evening, everyone. Myself, Jemima, pursuing MSc Applied Microbiology in Dwarkadas Govardhandas Vaishnav College. My topic on paper presentation is in vitro validation of antimicrobial activity of bioactive compounds from therapeutic plants. I'm under the guidance of Dr. M. Abhirami Ma'am, Assistant Professor of PG and Research Department of Microbiology. So, the contents are introduction, aim and objective, methodology, result, summary and conclusion. So, introduction. Basically, what we all know about medicinal plants. Medicinal plants, they have contributed continuous remedies for more than thousands of years. Comparing to the English medicine, normally they use many chemicals and all, but using the medicinal plants obtained from the therapeutic plants. For example, if we get cold, we'll rather than using tablets than taking antibiotics, our ancestors used to take that மிளகு பால் குடிங்க, like that they used to tell. that will be more efficient. That will be more efficient than using the antibiotics. So what we are using is medicinal plants. Medicinal plants, they are considered as the important source of nutrients because they have the higher therapeutic values. They are considered as the rich resource of ingredients which they can be further used in the development of drug. So among many plants, certain medicinal qualities of plants are selected, and they are believed to have that impacts in our day-to-day life, and they are very useful for our health and as well as to the environment. So the drugs derived from the natural source, that is drugs derived from the medicinal plants, play a major role in preventing the diseases and involved in the investigation of new drugs as they have no side effects. So these plants possess a variety of biochemical and pharmacological properties. So this review highlights the in vitro toxicity of the plant for the development of drugs. So what is the main objective of this paper presentation is the experiment is subjected to investigate the antimicrobial activity of the plants. So it is involved in detecting the presence of various bioactive compounds using different medicinal plants. So the main objective of this paper is to obtain the extract of medicinal plants using different solvents and to assess its antimicrobial activity. So, and then to characterize the presence of bioactive phytoconstituents and screening of those bioactive compounds. And the last objective is to perform in vitro assessment in clinical studies from the extract of medicinal plants. So, what are the medicinal plants that we are chosen is Eclipta alba, Catharanthus roseus, Carica papaya, Moringa oleifera. So, methodology. Coming to methodology, first step is the collection of different medicinal plants. So we are freshly collecting the medicinal plants from certain places, and the next step is preparation of plant extract using different solvents. The solvents used are acetone, methanol, chloroform, and using the different concentration of the solvents, the plant extract is prepared. And the third step is collection of microorganisms for antimicrobial activity. So how do we collect microorganisms? We put up the subculture for 24 hours, and the next day in the first day we inoculate the organism and we subculture it, and the next day we take up that and we subculture it and we incubate for 24 hours. So the fourth step is antibacterial activity. So the method used is agar-well diffusion method. As we all know, the basic method we use to assess the antimicrobial activity is agar-well diffusion method, which we use is MHC medium, that is Muller-Intenn agar. In that we load the antimicrobial extract that we extracted from the therapeutic plants in various concentrations, and then we swab the culture onto the medium and we incubate it for 24 hours. So the next day we check the results, and the another step we use is minimum inhibitory concentration, MIC. That is what is minimum inhibitory concentration? That is how lower the concentration of the antimicrobial extract we put up, that how much it inhibits the growth of the bacteria or whatever the microorganism. So methodology for further steps we used is phytochemical screening, thin layer chromatography, Fourier transform infrared spectroscopy, which is nothing but FTIR. So coming to phytochemical screening, which is nothing but phytochemicals. What are phytochemicals? Which is nothing but plant chemicals that are obtained from the plants. There is a potential plant extracts. So we are doing the phytochemical screening. The phytochemical screening involves flavonoids, terpenoids, saponins, alkaloids, tannins and oils. These are performed according to the procedure. So next it is thin layer chromatography. To separate the non-volatile liquids, thin layer chromatography is used. And the last step, Fourier transform infrared spectroscopy, is nothing but the functional group analysis. So these are the results. Preparation of plant extract. So in this, we check the antimicrobial activity, and methanol methanol showed the highest inhibitory zone, okay, and then antimicrobial activity of Eclipta alba. This is Carica papaya, and this is Catharanthus roseus. This is the result of MIC, phytochemical screening. This is the test for saponin, test for oil. So since we are using the 4 medicinal plants, these are the results. And in Carica papaya, using the methanol solvent, phytochemicals are all present in that. Thin layer chromatography. So in this, we have found out that these plants have antibacterial property, antifungal property, anti-cancerous property, and these can be used as the bioactive compounds from these plant extracts can be used for the further development of drugs.
In your presentation, you were elaborating more about the methodologies, but you had more results.
Yes.
So focus on results, okay? Because 5 minutes and 2 minutes for discussion. So within that time, okay, how far? Because you're all done so much, taken for the results, but you skipped. Okay, next. Instead, you are talking much about the methodologies. Okay, so focus on the results. Okay, so while explaining results itself, you can convey, they can understand. Okay, they are not MBA students. Okay, so it's a nice presentation. Thank you.
Our last presenter will be Kavya from Valyamal College.
Good evening to one and all present here. First of all, I thank to DG Vaishnav College for this giving me this wonderful opportunity. Myself, Kavya from 2nd MSc Biotechnology, Department of Biotechnology, Valyamal College for Women. My paper presentation is about screening, characterization and optimization of enzyme which is isolated from mangrove sediments. First of all, why I choose this mangrove forest? Because it is enriched with nutrients and trees and shrubs. It is an ecosystem which is a largely unexplored source of actinomycetes species. And actinomycetes species have the different properties such as it has ability to produce biologically active secondary metabolites and enzymes. It also screened for many anti-cancer, anti-tumor and antibacterial properties. Next. These are the various species of actinomycetes. This is a list. Next. Okay. Objectives and methodology of my study is first I have collected sample from mangrove forest, Pichavaram, Muttukadu Lake, Chennai, Tamil Nadu. Then I have isolated and identified the marine bacteria for various industrial enzymes. Secondary screening was done to 5 enzyme producing bacteria. Then check the quantitative assay, enzyme assay production for spectrophotometric assays. Then I have selected the best enzyme producers and its confirmation, enzyme confirmation. And then I have gone through optimization process to produce more enzymes, and partial purification was done by ammonium sulfate precipitation and dialysis process. Then protein profiling was identified by using SDS page and zymogram analysis. And finally, the selected culture was subjected to molecular characterization, DNA isolation and gene sequencing. Next. Okay, collection of sample. The sediment sample which I have collected was gone through preheat treatment. Then I have isolated the culture. The media preparation was starch casein agar, ISP 5 agar, and then marine nutrient agar. Filtered seawater 50% and deionized water 50% were used to cultivate the media. Okay.
The cultivated media was stored at 28 degrees Celsius for 3 weeks. Then these are the location I have collected the sample. The 24 isolates were isolated from the sample I have collected. These are the pictures of 24 isolates. Okay. The 24 culture isolates which I have isolated was subjected to enzyme production media to determine the enzyme activity. The enzyme which I have chosen was starch, lipid, gelatin and cellulose. The results were observed by adding solutions to the culture plate. For gelatinase, it's ammonium sulfate. For lipase, it is a direct observation. For cellulose, it is a congruent dye. For starch, it's a Gram's iodine. Next slide. These are the results of enzyme activity. Okay, the selected 5 culture isolates based on the further results. Then I have selected R11 for showing more enzyme activity. I have done enzyme assay based on spectrophotometric assay. Gelatinase and cellulase enzymes were done. On both the enzyme assay, I have concluded that R11 was showing more enzyme activity on both the enzymes. Next slide. Next, I have gone through optimization process. For optimization, I have gone through carbon source, nitrogen source, pH and temperature for determining the best enzyme producer. From carbon source, glucose shows more activity. Then nitrogen source, yeast source, the more activity. pH at pH 8 and the temperature 35 degrees Celsius, the enzyme shows more activity. Next slide, please. Then partial purification was done through ammonium sulfate precipitation. 70% of ammonium sulfate precipitation was done. And then the supernatant was discarded by centrifugation process of 6000 rpm for 10 minutes. Then the pellet was collected. The enzymes were seated under the pellet. Then the pellet was collected and gone through dialysis process to remove the salt concentration and some other organic molecules. Next slide. Then I have selected to further process for protein profiling by using SDS page and zymogram analysis. These are the results. SDS page was determined to know the molecular weight and zymogram analysis was done to enzyme activity. Then the selected culture was subjected to DNA isolation by using agarose gel electrophoresis method, and 16S rRNA gene sequencing was done to determine the bacterial isolate. Next slide, please. From this study, I have concluded that the gram-negative agrobacterium intermedium and agrobacterium species was chosen more enzyme, cellulase enzyme activity. But why I choose actinomycetes? Because I like to work on actinomycetes, but the result I have got is agrobacterium, a gram-negative species. Thank you.
First, okay, it's a nice presentation. So, where you completed all this work? In Valiamal College or you did any
private labs?
Okay, okay, fine. So in that presentation, you mentioned like collection photos, collection of samples. Why you didn't include the screening photos? The enzyme screening, right? You have used 4 enzymes, right? Why you didn't include that photos?
Slides will be moved, so I do a flow chart and show the results and positive, negative.
See, in that case, that picture plays major because isolation anybody can do. Okay, but you screened means you have to include those photos in which gives positive, because this is not like a viva. You are here for a presentation, so everyone should see, okay, if because from this they can learn many things by using those medium, by using those methods. Okay, this will be like this. So that screening plates are very, very essential for the presentation. Okay, and yes, and you mentioned that cymography, right?
Yes, sir.
Okay, so is it done by you or what?
Yes, sir, done by me.
Okay, thank you.
Thank you, sir.
I, on behalf of PG and Research Department of Microbiology, would like to thank our judge, Dr. DJ Mukesh Kumar, for taking time from his busy schedule. Your time and dedication made this event possible, sir. Thank you very much. Thank you, sir.
The valedictory speech will begin in few minutes. I request you all to maintain silence.
அது அவங்களோட ஃபேஸ் அவ்வளோ கிளியராக இருக்காது. கேட்கும், பேசுறது கேட்கும். ஆக்சுவலாக யூடியூப் சேனல் இருக்குது, அந்த டிஜிவிசி மைக்ரோ, அதில் நேத்தோட ப்ரெசன்டேஷன் சர்ச் பண்ணி பாருங்க. டிஜிவிசி மைக்ரோ. இருக்கா? சேனல் ஆஃப் பண்ணிட்டு ஓடிட்டு தான் இருக்கு. விஷாக்கா கொடுத்து
ரைட்.
సుందర్ సుందర్ గారు லேப்டாப் இருக்கு கீழே வச்சு
Good evening, everyone. I would like to welcome you all for the valedictory function of International Conference Next Generation Microbiologist Challenges and Opportunities 2.0. Now, I take great privilege in welcoming Dr. M. Abhirami, Assistant Professor, Department of Microbiology, to welcome the gathering.
Good evening, everyone. It gives me a great pleasure to deliver the welcome address for the validation of our 2 days international conference on Next Generation Microbiologist Challenges and Opportunities 2.0. We have gathered here today to celebrate the successful event of this year, which has been intellectual for all of us. On behalf of the PG and Research Department of Microbiology, it's my privilege to welcome our distinguished secretary, Dr. Ashok Kumar Mundraji, our principal, Captain Dr. S. Santosh Babu sir, and our chief guest of the day, today's function, Dr. Prabhu, Department of Microbiology, University of Madras, to this special gathering. Our beloved secretary, Dr. Ashok Kumar Mundraji, we are grateful for their support, and our beloved principal, Dr. Captain S. Santosh Babu sir, we are grateful for their unwavering commitment to education, and their leadership has been inspiration to all of us. We are honored to have such a distinguished chief guest, Dr. Prabhu sir, and your achievements and contribution in the field of microbiology are truly remarkable, and it's a privilege to have you here. It's my great honor to welcome our resource person, Dr. Ganesh, ICMR, Chennai. It's my great honor to welcome Dr. P. Vidya, the head Department of Microbiology, the organizing secretaries, the research scholar, and my students, and the dear participants to this patriotic functions of international conference. This conference was an opportunity for us to exchange the ideas, experience, and knowledge. It has served as a platform for experts and professionals to come together and share their insights on important topics of global significance. I hope that you all had a wonderful time engaging in stimulating discussions and learning from one another. Let us now take a moment to reflect on the achievement of this conference and the impact it will have on our respective fields. I would like to extend my gratitude to all the speakers and participants for making this event a huge success. Once again, I extend my gratitude to all. Welcome you all.
May I now request Dr. P. Vidya, Head Department of Microbiology, to honor our chief guest, Dr. Prabhu, Head Department of Microbiology, Dr. ALM PG IPMS, University of Madras, Taramani, with a memento and a bouquet as a token of love and appreciation. I now call upon Ms. Sharmila, Research Scholar, Department of Microbiology, to introduce our chief guest, Dr. Prabhu, Head Department of Microbiology, Dr. ALM PG IPMS, University of Madras, Taramani.
Respected dignitaries on the dais and off the dais, faculty staff and my dear students. Good evening, one and all gathered here. It's my immense pleasure to introduce our chief guest for this valedictory function of International Conference on Next Generation Microbiologist Challenges and Opportunities 2.0. We are honored to have you, sir, Dr. Prabhu, Head of the Department of Microbiology, Dr. ALM PGIBMS, University of Madras, Tharmani. He did his Masters in Environmental Microbiology on isolation of Cryptococcus neoformans from living trees in Guindy National Park, and his doctorate in Medical Microbiology on molecular characterization of Group C and Group C streptococci. He worked as a project fellow in ICMR funded project during the year of 2006 to 2007. Sir has worked as a research fellow in the Department of Microbiology, University of Madras during 2007 to 2008. Dr. Prabhu upgraded from research fellow to senior research fellow in the Department of Microbiology, University of Madras during 2009. He served as a teaching cum research fellow TRF in the Department of Microbiology, University of Madras during 2010. He worked as a team-led clinical research in TCS Life Science during 2010 to 2014. He is promoted as assistant professor in the Department of Microbiology, University of Madras during the year of 2014. Currently, Dr. Prabhu is working as a head in charge Department of Microbiology, University of Madras. He has published around 20 papers in peer-reviewed international journals. To name a few, Journal of Clinical Microbiology, Trends in Biomedical, Asian Public Journal of Health and Science, and Journal of Global Science. He has been awarded with Teaching cum Research Fellow TRF awarded by University of Madras, Senior Research Fellow SRF awarded by Department of Biotechnology, Government of India, Research Fellow awarded by University Grants Commission. He has completed 5 projects. Sir is currently guiding 4 PhD students. Thank you, sir, for coming here in spite of your busy schedule. Once again, we welcome you, sir.
Respected Chief Guest, Dr. Prabhu, Head Department of Microbiology, University of Madras, IBMS, respected Secretary, beloved Principal who is not here, I just convey my wishes to him, and staff, students of DGVC, participants and my dear students, I, Dr. Kritika, on behalf of PG and Research Department of Microbiology, feel honored to present the summary of our International Conference Next Generation Microbiologists Challenges and Opportunities 2.0. On account of Silver Jubilee celebration, the department has planned to organize various curricular, co-curricular and community service events throughout the year, and this international conference is our 17th event. Microbiology is at a turning point in its 120-year history. Microbiology is still a young science, so there are many frontiers yet to explore. Next generation microbiologists need to integrate several scientific disciplines in the early stage of their careers to keep up with the ever-changing nature of microbiology and to ensure timely retort to the current global health and biodiversity threats. In order to edify and invigorate budding microbiologists, we, the Microbiology Research Department, selected the theme for our international conference. The conference was initiated in the September 2022 on discussion with the advisory committee members. The committee finalized the theme covering the various fields of microbiology as thrust areas. The conference preparation was started with a call for abstracts. 80 abstracts were received from various colleges of Tamil Nadu and 2 abstracts from South Korean University. All the abstracts were accepted to encourage the young researchers. The abstracts were categorized as oral and poster presentations, and the conference proceedings was released during the inaugural session. The conference brought together 180 participants from 20 institutions of Tamil Nadu. The ultimate objective of the conference is to share experience, inspire people, build network, and bridge the gaps in different avenues of microbiology. The conference was inaugurated by Dr. K. Kadirvelu, Joint Director, DRDO Industry Academia Center for Excellence, Bharatiyar University. He appreciated the department for organizing such event and he expressed a willingness to arrange funds through DRDO to strengthen our research program. Dr. Prabhu Devasigamani, CEO, Diligence Bio, Puducherry, delivered the keynote address. He emphasized that career opportunities exist in various sectors at different levels for microbiology students. The inauguration was followed by a technical session. The first session was shared by Professor Dr. S. Ganapathy Venkatasubramanian, Department of Environmental Law and Management, Anna University. He enlightened the participants on effective environmental management through environmental legislations. He clearly explained about the environmental management system, EMS, and environmental law, government, and public policy towards the environment. Second session of day 1 was handled by Dr. Anand Anbarasu, Professor, Department of Biotechnology, VIT, Vellore. He discussed about the future challenges and opportunities of modern microbiology in bioinformatics perspective. He highlighted the techniques, next generation sequencing and its application in medicine, along with the global opportunities in the field of bioinformatics. Mr. Jeevanand Raju, Hygiene Auditor, Abu Dhabi, shared the first session of second day through online mode. In his presentation, he offered suggestions on the new era of smarter food safety blueprints. Dr. Harish Sri Chandramurthy, Vice Chairman, Center for Stem Cell Research, King Khalid University, Saudi Arabia, our second speaker for the day 2, shared the highlights of recent trends in stem cell research and emphasized opportunities in the field of stem cell. The last technical session was handled by Dr. B. Ganesh, Scientist E, Deputy Director, Laboratory Division, ICMR, National Institute of Epidemiology, Chennai. He clearly explained about the infectious agents directed in the worldwide and also the national strategy on infectious diseases cutting across all the relevant sectors. And now we are in the final session of our conference. We thank Dr. D. Prabhu, Head, University of Madras, IBMS campus, accepting our invitation to deliver the valedictory address. Hope the conference is a fruitful experience for all of you. I express my sincere thanks for joining us and making this event a grand success. Thank you.
Now, I request the Chief Guest, Dr. Prabhu, Head Department of Microbiology, University of Madras, to enlighten us with few words.
Thank you. So அவங்களே சொல்லிட்டாங்க few words ன்னு சொல்லிட்டு ஸோ அதனால இட்ஸ் ஓகே. ஸோ தமிழ் தெரியும் இல்லை எல்லாருக்கும்? தமிழில் பேசலாம் இல்லையா? அது ஏன்னா அதுதான் புரியும். ஏன்னா காலையிலேருந்து நேற்றுலேருந்துலாம் இங்கிலீஷாக கேட்டுட்டு போய் எனக்குமே அது ஒரு 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 இரிட்டேட்டிங் லாங்குவேஜ் தான் சொல்லுவேன். பட் என்ன பண்ணுறது, நம்ம வேலை அந்த மாதிரி இருக்கிறதுனால நம்ம இங்கிலீஷில் பேச வேண்டியதாக போச்சு. அது ஒரு 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 ஸ்டேட்டஸ் ஆகிடுச்சு இல்லையா? ஸோ அதனால சொல்கிறேன். ஸோ தேங்க்யூ வெரி மச் ஃபார் த குட் இன்ட்ரோடக்ஷன். Somebody was giving, right? So for the good introduction, and first of all, it is my honor, right? To talk before you. So first, thanks and sollikren. So I would like to thank the management, DG Vaishnava College, principal, and Madam Vidya, and Madam Kritika, Madam Jagdishwari, and other faculty members, right? For inviting me for this valedictory function. So நான் நிறைய டைம் எடுக்க விரும்பல, ஜஸ்ட் ஒரு 2-3 இம்பார்ட்டன்ட் விஷயம் மட்டும் நான் சொல்லிடலாம். ஸோ the topic is very good. I can say is need of an hour. I can say the challenges and opportunities. பட் இந்த டாபிக்லேயே வந்து பார்த்தீங்கன்னா 2 விஷயம், அதாவது நீங்கள் 2ஆக பிரிச்சுக்கலாம், challenges and opportunities. So as a microbiologist, I can say challenges will be like a pre-COVID era. Opportunities are like a COVID era. So pre-COVID era வந்து பார்த்தீங்கன்னா அந்த அளவுக்கு we don't have that much challenge. I mean opportunities for the microbiologists are lab அப்படிதான் சொல்லுவாங்க. But of late, you're going to see microbiologists, oh my God, you people have done a great job. I'm saying that you are the frontline healthcare worker. That's all. The scene has been recreated, I can say. So இதை ஏன் சொல்ல வரேன்னா, இப்போ வந்து பாத்தீங்கன்னா நம்மளுக்கு ஆப்பர்ச்சுனிட்டிஸ் என்ன பொறுத்த வரைக்கும், this is my view, I'm not sure what the other speakers and guests they were talking about, but என்ன பொறுத்த வரைக்கும் வந்து I will say that we have opportunities now. Only thing is you should know where to tap these opportunities. And see, இப்போ இன்னைக்கு பார்த்தீங்கன்னா ஆல்மோஸ்ட் ஒரு 6-7 ஸ்பீக்கர்ஸ் வந்திருப்பாங்களா? 7 ஸ்பீக்கர்ஸ் வந்திருந்தாங்க. எத்தனை பேர் உங்களுடைய சிவி உங்களுடைய ஐடியாஸ் சொல்லியிருக்கீங்க? ப்ளீஸ் ரைஸ் யுவர் ஹேண்ட்ஸ். இல்லை உங்கள் சிவி அட்லீஸ்ட் சார் என்னோட ஐடியா உங்களோட ஐடியா மேட்சாக இருக்கு, ஒய் டோன்ட் வி என்ன சொல்றது, நான் உங்களோட இது ஐ மீன் உங்க ப்ராஜெக்ட்ஸ்லயோ இல்லை உங்களோட இண்டஸ்ட்ரி ஆர் லேப் எத்தனை பேர் கேட்டீங்க? ஸோ எஸ் ஒன். நீங்க இன்னொருத்தருங்களா? ஓகே ஃபைன், சாரி. ஸோ 2 பேர் தான். ஸோ ஏன் why I'm saying is ஆப்பர்ச்சுனிட்டிஸ் கொடுத்தாங்க, பட் நம்ம யூஸ் பண்ணிக்கல, அவ்வளவுதான். See, this is what, see, being a teacher, we can show the way, right? இப்படி போனா நல்லா இருக்கும்னு தான் சொல்ல முடியுமே தவிர உங்களுக்கு see you people are not like a school kids. என்ன சொல்றது, வாங்க போய் கொண்டு போய் உங்களை வந்து விட்டு அதான் ஸ்கூல் வரைக்கும் போய் விட்டுட்டு வரேன் அப்படின்ற மாதிரி சொல்ல. see, these are all the opportunities, right? so challenge வந்து பாத்தீங்கன்னா inhibition. உங்ககிட்ட இருக்கிற inhibition இருக்கு பாத்தீங்களா, that is a challenge. போய் கேட்கலாமா, கேட்டா என்ன நினைப்பாங்க, நத்திங் ராங். என்ன சொல்ல போய் கேட்கறதுனால என்ன உங்களை திட்டவா போறாங்க, இல்ல இல்லையா, ரைட்? ஸோ மேடம் வந்து பாத்தீங்கன்னா ஒரு ஆப்பர்ச்சுனிட்டி கிரியேட் பண்ணாங்க. ஸோ அதை நீங்க யூட்டிலைஸ் பண்ணிருக்கலாம், தாராளமா யூட்டிலைஸ் பண்ணிருக்கலாம். நத்திங் ராங் இன் தட் கோயிங் அண்ட் ஆஸ்கிங் தெம் சார் இஸ் இட் ஒரு மேபி ஒரு பர்மிஷன் கேட்டிருக்கலாம் அவங்க, சார் இஸ் இட் ரைட் டைம் டு ஸ்பீக் டு யூ அபவுட் மை இன்டர்ன்ஷிப். ஸோ எடுத்தவொடனே ஜாப்னு கேட்டாலும் யாருமே வந்து தே வில் நாட் அக்செப்ட். அவர் எடுத்தவொடனே போய் சார் நான் வந்து உங்ககிட்ட பிஹெச்டி ஜாயின் பண்ண போறேன்னு சொல்லி கேட்டாலும் யாருமே வந்து அக்செப்ட், இன்க்ளூடிங் மீ, ரைட்? ஒரு approach is going to be very important. Maybe you should have asked, sir, is there any chance where I can do internship project? புரியுதுங்களா? அந்த மாதிரி நீங்க கேட்டு ஒரு அப்ரோச் பண்ணி போறீங்கன்னா that is going to be an opportunity. see எனக்கு ஒரு ஆப்பர்ச்சுனிட்டி அது மாதிரி கிடைச்சது ஆக்சுவலி. ஸோ ஃபார் மீ லைக் frankly speaking என்ன சொல்றதுன்னா, நம்ம டீச்சிங் ஜாபே வரக்கூடாதுன்னு ஒன்று, எனக்கு பிடிக்காத ஒரு ஜாப் வந்து பாத்தீங்கன்னா டீச்சிங் ஜாப் தான், சரிங்களா? வரக்கூடாதுன்னு இருந்தேன். அதுக்கும் மேல வந்து பாத்தீங்கன்னா மைக்ரோபயாலஜின்னு ஒரு சப்ஜெக்ட் எனக்கு தெரியாது, எதுவுமே எனக்கு சீட் கிடைக்கல. ஐ டென்ட் கெட் எனி சீட். நான் ஒரு ஆவரேஜ் ஸ்டூடெண்ட். என்னடா அப்படி என்னையே நான் தாழ்த்தி சொல்றேன், அதுதான் உண்மை. ப்ராக்டிகலி ஐ எம் ஜஸ்ட் டெல்லிங் யூ தட் ஐ ஜஸ்ட் கேம் ஜஸ்ட் லைக் தட் பிகாஸ் ஐ டென்ட் கெட் மெடிக்கல் சீட் கிடைக்கல, எம்பிபிஎஸ் கிடைக்கல. ஸோ எம்பிபிஎஸ்க்கு ஈக்குவலா என்னன்னு பாத்தீங்கன்னா ஒரு 20 years முன்னாடி வந்து பாத்தீங்கன்னா ஓகே மைக்ரோபயாலஜி பயோகெமிஸ்ட்ரி அந்த மாதிரினாங்க. ஸோ ஐ ஜஸ்ட் ஜாயின் பச்சவாஸ் காலேஜில் தான் நான் படித்தேன். நான் பிஎஸ்சி மைக்ரோபாலஜி அங்கே தான் படித்தேன். அதுக்கப்புறம் வாட் இஸ் ஈவினிங் காலேஜ், பட் ஆனால் டேல எடுத்தாங்க. ஏன்னா டேலே ஒன்றும் பண்ண மாட்டோம், ஈவினிங்ல பண்ண என்ன பண்ணுவான்னு பாருங்க அப்படின்னு சொல்லிட்டு டேல எடுத்தாங்க. அது வேற விஷயம் வைங்க, அட்ஸ் அ டிஃப்ரெண்ட் ஸ்டோரி. அண்ட் தென் லைக் அதுக்கப்புறம் ஐ டோன்ட் நோ வாட் டு டூ. அப்போ வந்து பாத்தீங்கன்னா ஐபிஎம்எஸ் இன்ஸ்டியூட் ஆஃப் பேசிக் மெடிக்கல் சயின்ஸ் அப்படின்னு சொல்லி எல்லாருக்குமே அது ஒரு கனவு ஆக்சுவலி. இப்போ மைக்ரோபாலஜி படிக்கிறவங்க மேபி உங்களுக்கு தெரியுமான்னு தெரியல, மேடம் கிட்ட கேட்கலாம், அதர் ஃபேக்கல்டிஸ் கிட்ட கேட்கலாம், அது ஒரு கனவு மாதிரி இருந்தது. அப்போ சொல்றேன் நான். ஸோ ஐ டென்ட் என்ன சொல்றேன், இப்போ ஒரு ஐபிஎம்எஸ்ஸா ஓகே ஜஸ்ட் ஒரு என்ட்ரன்ஸ் எழுதுனேன். என்ட்ரன்ஸ் எழுதி எனக்குமே அது எல்லாம் எழுத சொல்றாங்களேன்னு எழுதுனேன். பட் எனக்கு எப்போ ஒரு ஆப்பர்ச்சுனிட்டி அப்படின்னா, ஸோ ஐ வாஸ் மீட்டிங் ஒரு ப்ரொஃபஸர், பேர் சொல்ல விரும்பல, என்ட்ரன்ஸ் எக்ஸாம் அப்போ. ஸோ தட் வாஸ் லைக் எ ட்ரிகர் பாயிண்ட் ஃபார் மீ. ஸோ ஆல்மோஸ்ட் லைக் ஒரு இல் ட்ரீட்மெண்ட் மாதிரி தான், நீ எல்லாம் படிச்சு என்ன ஆக போறேன், ரைட்? பிகாஸ் நம்ம வந்து அந்த பர்சனாலிட்டி டெவலப்மெண்ட்டுங்கிறது வந்து பாத்தீங்கன்னா அதுக்கப்புறம் தான் பிஹெச்டி முடிச்சு ஒரு கார்பரேட் போயிட்ட பிறகு தான் ஒரு பர்சனாலிட்டி டெவலப்மெண்ட் எல்லாம். அதுக்கு முன்னாடி வந்து பாத்தீங்கன்னா ஒரு மாதிரி ரகடா இருப்போம் நாங்க. அப்போ வந்து நீ எல்லாம் அப்படி. ஸோ தட் வாஸ் அ பாயிண்ட், ரைட்? ஸோ இதுக்காகவே என்ன ஆச்சுன்னா ஐ வென்ட் டு என்ன சொல்ற கிடைச்சிருச்சு, அது வேற விஷயம் சொல்லு. ஸோ இப்படி ஒரு ஆளு இப்படி ஒரு டிபார்ட்மெண்ட்ல இருக்காரு, நான் வந்து எம்எஸ்சி படிக்கிறேன்னா அது கட்டாயம் ஒரு டக் ஆஃப் வரும். ஸோ அதுக்கு முன்னாடி நான் என்ன வந்து ஐ நீட் டு டெவலப் மை ஸ்கில் செட்ஸ், ரைட்? ஸோ அதுக்கு ஒரு சின்ன லேப்ல வந்து ஐ வென்ட் தேர் அஸ் ஒரு இன்டர்ன், ஒரு நைட் டியூட்டி மாதிரி, ஆல்மோஸ்ட் லைக் ஒரு நைட் டியூட்டி மாதிரி தான். ஸோ ஐ லேர்ன் மை என்ன சொல்றது மைக்ரோபயாலஜி. மேக்ஸிமம் வந்து அங்கே பச்சவாஸ் காலேஜில் ஒன்றும் இல்லை, எல்லாருக்கும் தெரியும் ஏன்னு சொல்லிட்டு அதை பற்றி நான் பேச விரும்பல. ஸோ அங்கே நான் படித்தேன், எக்ஸ்பீரியன்ஸ் கெயின் பண்ணேன். ஸோ ஐ வாஸ் ஆல்மோஸ்ட் லைக் என்ன சொல்றது நல்ல ஒரு குட் ஆன்ஸ். அப்போதான் எனக்கு ஒரு விஷயம் புரிஞ்சுது. ஸோ ஆப்பர்ச்சுனிட்டி வந்து கிரியேட் பண்ணணும்னா அது நம்ம கிரியேட் பண்ணிக்கணும் ஒன்று, இன்னொன்று வந்து யாராவது கிரியேட் பண்ணி கொடுத்தா அது யூஸ் பண்ணிக்கணும். ஸோ யூஸ் பண்றது எப்படின்னா இட்ஸ் நாட் லைக் தட் ஓகே. நான் பார்த்துருக்கேன் நிறைய பசங்க ஐ டிட் மை ப்ராஜெக்ட் இன் ஐஐடி, I did my project in project dissertation போறப்போ சொல்லுவாங்க, it's not about doing project in Premier Institute. It is about like the knowledge you are going to gain from the project, எங்க வேணாலும் பண்ணலாம். நீங்க டிஜி வைஷ்ணவலே வந்து ப்ராஜெக்ட் பண்ணிட்டு whatever the knowledge you are going to gain, that is going to be useful. rather than வந்து நான் ப்ரீமியர் இன்ஸ்டியூட்ல பண்ணேன், இங்க பண்ண ஐபிஎம்எஸ்ல பண்ணேன், ஐஐடில பண்ணங்கிறதுலாம். ஏன் இது சொல்ல வரேன்னா, opportunities will come to you when you have a good knowledge on the domain. Right. So challenges ஒரு பக்கம் இருக்கட்டும். ஏன்னா அந்த ஆப்பர்ச்சுனிட்டி கிடைக்கிறது ஒரு சேலஞ்சு. பிகாஸ் நாட் ஆல் த பீப்புள் வில் கெட் ப்ராஜெக்ட் இன்டர்ன்ஷிப் இன் ஐஐடி ஆர் ஐபிஎம்எஸ் ஆர் வாட் எவர். என்ன கூட சொல்ல, தட்ஸ் ஆல்சோ ப்ரீமியர் இன்ஸ்டியூட் ஐ கேன் சே. இப்போ என்ன எல்லாம் பார்த்தீங்கன்னா பயங்கர டிமாண்ட். சார் சொல்லிட்டு இருந்தாரு, சார் ஆயிரக்கணக்கான அப்ளிகேஷன்ஸ் வருது, ஒன்லி வி ஆர் செலக்டிங் 2. லைக் தட்ஸ் ஹவ் லைக் மேக்கிங் த ப்ராசஸ் என்ன சொல்றது கொஞ்சம் நல்லா இருக்கணும் அப்படின்னு சொல்லிட்டு. ஏன்னா எல்லாருமே எடுத்தோம்னா குவாலிட்டி வில் கோ ஆஃப். ரைட். ஸோ ஆப்பர்ச்சுனிட்டியை நீங்க கிரியேட் பண்ணுங்க. அதர்வைஸ் இந்த மாதிரி ஒரு மேடம் ஒரு ஆப்பர்ச்சுனிட்டி கிரியேட் பண்ணாங்கன்னா ட்ரை டு யூட்டிலைஸ் தட் ஆப்பர்ச்சுனிட்டி அண்ட் ஜஸ்ட் கோ ஃபார் இட். ராதர் தென் என்ன சொல்றது ஒரு கான்ஃபரன்ஸ் வந்தோம், ஒரு செஷன்ஸ் அட்டென்ட் பண்ணோம், தட்ஸ் இட், லைக் வந்து போகணும் அப்படிங்கிற மாதிரி வேண்டாம். ரைட். லைக் ஆல் திஸ் கான்ஃபரன்சஸ் ஆர் for 2 things. One is sharing the knowledge, and second thing is like getting the contacts. Contacts are very important. நம்ம ஒரு படத்துல செந்தில் சொல்வார் தெரியுங்களா, இன்ஃபர்மேஷன் இஸ் வெல்த் ன்னு சொல்லிட்டு. Conferences are something like that. So whatever the information you are going to gain, that is going to be very helpful for your career. That is going to be like ஒரு நல்ல ஒரு கான்ஃபரன்ஸ் ஆல்வேஸ் சொல்ல will be a turning point for many people. Right. So அந்த மாதிரி இந்த கான்ஃபரன்ஸ் உங்களுக்கு இருந்திருக்கும்னு நான் நினைக்கிறேன். So ஆப்பர்ச்சுனிட்டிஸ் பாத்தீங்கன்னா நிறைய இருக்கு. இப்போ பயோபிளாஸ்டிக்ஸ், ரைட், பயோபிளாஸ்டிக்ஸ், பயோபெஸ்டிசைட்ஸ், இண்டஸ்ட்ரியல் மைக்ரோபயாலஜி, ஃபுட் மைக்ரோபயாலஜி இஸ் பூமிங் லைக் எனிதிங். ஸோ பீயிங் அ மெடிக்கல் மைக்ரோபயாலஜிஸ்ட், நான் வந்து இன்னொரு இது வந்து சொல்லக்கூடாதுன்ட்டு இல்லை, பட் this is a practical truth that I want to say is like industrial microbiology and food microbiology is booming like anything. And innovations, so just like that opportunity பத்தி நம்ம பேசுறப்போ வந்து innovation பத்தி சொல்லாம விட்டோம்னா I don't think so. Like I'm just doing justice for this validatory function. Innovations. So ஒரு விஷயம் சொல்லுவாங்க. So for example, innovation, what is innovation சொல்லிட்டு கேட்டுருப்பாங்க. Why innovation? Why என்ன சொல்றதுன்னா ஏன் வந்து அதை பத்தி நம்ம பேசணும், ஏன் வந்து அது இம்பார்ட்டன்ட் அப்படின்னு சொல்றேன்னா, எஸ், யுவர் நெசசிட்டி ஐ கேன் சே. See for example என்ன சொல்றதுன்னா, you need something, like for example plastic issue இருக்குது, பாலிமர் synthetic பாலிமர் இருக்கிறதுனால நம்மளுக்கு நிறைய ப்ராப்ளம் இருக்கிறதுனால தான் we are talking about bioplastics, we are talking about biopesticides, biodegradation, right? So இந்த மாதிரி எல்லாம் வந்து பாத்தீங்கன்னா நெசசிட்டி இருக்கிறதுனால தான் வந்து yes, it is just like that booming like anything. உங்களுக்கு தெரியுமா இல்லைன்னு தெரியல, இந்த பயோபிளாஸ்டிக்ங்கிற கான்செப்ட் வந்து இந்தியாவுக்கு வேணா அது புதுசா இருக்கலாம், பட் I think Western and Eastern countries started developing. So who is working on this? The microbiologists like you people. I think the scope is more for you people than the other, even the IT people எல்லாம் கூட வந்து பாத்தீங்கன்னு நினைச்சீங்களேன், ரெசெஷன் வீட்டுக்கு அனுப்பிச்சு விட்டுருவாங்க. ஆனா அப்படி என்ன சொல்ற மைக்ரோபயாலஜிஸ்ட் பொறுத்த வரைக்கும் பாத்தீங்கன்னா இட் இஸ் லைக் ஒரு கோல்டன் ஆப்பர்ச்சுனிட்டி கூட நம்ம சொல்ல முடியாது. இட்ஸ் அ கைண்ட் ஆஃப் ஒரு டாக்டர்ஸ்க்கு ஈக்குவலான ஒரு போஸ்ட்னு சொல்ல. ஏன்னா டாக்டர்ஸ்க்கு வந்து பாத்தீங்கன்னா ரிட்டையர்மெண்ட் கிடையாது. At the same time, even for the microbiologists, like there's no retirement, right? Even at the age of 80, right? Like நீங்க போய் மைக்ரோஸ்கோப் பார்க்கலாம். You can go for என்ன சொல்றது ஒரு பிளேட் ரீட் பண்றதுக்கு, இல்ல ஒரு கல்ச்சர் இது பண்ற ப்ராசஸ் பண்றதுக்கு you can go to a lab. See, that's a different story. என்ன சொல்றது காசு கொடுக்குறாங்க இல்ல கம்மியா நிறைய. See what I heard from other people, right? I say what sir, like after finishing MSc the pay they are giving is very meager. But for your information, even if you're going to finish information technology or a computer science, the pay is going to be same. Only thing is என்ன சொல்றதுன்னா நம்ம ரொம்ப சினிமேட்டிக்கா இருக்கிறோம். ஏன்னா சினிமால தான் வந்து பாத்தீங்கன்னா அபி என்ன சொல்றது ஒயிட் காலர் ஜாப், ஐடி. ஏன்னா நம்ம படத்துல எல்லாம் அப்படிதானே காட்டுறாங்க. லாட் ஆஃப் கேர்ள்ஸ், ஒரு பையன் ஒரு எப்ப பார்த்தாலும் கேப்டீரியா, அப்படிலாம் ஒண்ணுமே கிடையாதுங்க. நீங்க ஐடி கம்பெனிக்கு எல்லாம் போனீங்கன்னு நினைச்சீங்கன்னா சம்திங் டிஃப்ரெண்ட். பிகாஸ் ஐ எம் ஃப்ரம் கார்பரேட் பேக்ரவுண்டுங்கிறதுனால சொல்றேன், வேலை அப்படி செய்வாங்க. பட் சினிமால வந்து அப்படி காட்டுறாங்க, நிறைய சம்பாதிக்கிற மாதிரி காட்டுறாங்க. நோ, ஐடி பீப்புள் ஈவன் என்ன சொல்றது they are getting equal whatever the pay you are going to get as a first salary. So don't think that like your job is very very என்ன சொல்றது ரொம்ப கீழே அப்படின்னு நினைச்சுக்காதீங்க. So your job is equivalent to the other professions also. And in fact I will say that like microbiology courses professional is, like almost it is superior than any other post. And as I told you before that it is equivalent to a medicine which is a life saving, because I think 2 days back I was just giving a talk about opportunistic infections, where like I was just telling them, saying based on your report, like a patient is going to get treated, right? And whatever the report you are going to give, that is going to be like very very important for the patient. So rather than like working for the, see, I'm not saying that monetary aspect is sort of the least important. Monetary aspect is important, ஒரு criteria தான் நான் சொல்றேன். But apart from that, like you should also be like like அந்த aesthetic touch ன்னு சொல்ல கலோக்கியலா சொல்ல சயின்டிஃபிக்கா சொல்ல மாட்டாங்க, aesthetic, that you are also a human being. So that's the reason we'll say that like when it comes for the சொல்ற ஒரு ட்ரீட்மெண்ட்டா இருக்கட்டும், இல்லாட்டி ஒரு மெடிசனா இருக்கட்டும், இல்லாட்டி ஒரு ஃபுட் மைக்ரோபயாலஜி இண்டஸ்ட்ரியா இருக்கட்டும், வாட் எவர் த ரிப்போர்ட் யூ ஆர் கோயிங் டு கிவ் இஸ் வெரி வெரி இம்பார்ட்டன்ட். இஃப் யூ கோயிங் டு ஃபுட் இண்டஸ்ட்ரியில போயிட்டு குவாலிட்டி கண்ட்ரோல்ல வந்து நீங்க வந்து பரவாயில்ல போட்டு விடுங்க, டெஸ்ட் பண்ணாம கொடுங்க. ஒரு காலத்துல அப்படிதான் சிங்க் டெஸ்ட்னுவாங்க. ஐ திங்க் டாக்டர் சுந்தரராஜன் சார் சொல்லுவார், அந்த சிங்க் டெஸ்ட்னா என்ன பண்ணுவாங்கன்னா, ஒரு பர்டிகுலர் ஆளுங்க, ஐ டோன்ட் வாண்ட் டு மென்ஷன் த பீப்புள், ரைட், பிளட்டை கலெக்ட் பண்ணிட்டு சிங்க்ல ஊத்திட்டு பாசிட்டிவ் நெகட்டிவ் கொடுப்பாங்க. 15 years 20 years டாக்டர் சுந்தரராஜன் என்னோட ப்ரொஃபஸர் அவர் சொல்லுவாரு, ரைட். அந்த மாதிரி இல்லாம, ஸோ தெர் இஸ் ஆல்வேஸ் என்ன சொல்றது, தெர் இஸ் ஆல்வேஸ் யூ ஷுட் feel proud that like you are a microbiologist, and there are a lot of opportunities. Forget about the challenges are very 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 meager, very 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 meager. And after COVID, you won't believe that like people, those are very good background of experience. That's what I was telling you, that gain knowledge. If you have a good knowledge after finishing like MSc, right, they were getting almost 30,000 bucks per month. After COVID, there are so many labs, like they were just struggling to get good microbiologists. We have microbiologists. I was telling that like almost ஒரு 30,000-40,000 பேர் வர இந்தியா ஃபுல்லா. But why still there is a huge demand? Why there is still like unemployment among the microbiologists? The thing is like we are not developing the skills. So opportunity will come to you unless you need to develop the skills. So skills develop பண்ணலனா, I don't think so. See, don't get the degree. So mere என்ன சொல்லுங்க, parents சொல்றனால எல்லாம் ஒரு ambition, ஒரு vision, ஒரு mission, don't think so. நான் இப்போ சொல்றேன், இதெல்லாம் அட்வைஸ் எல்லாம் கிடையவே கிடையாது, சரிங்களா? ஜஸ்ட் a suggestion. இப்படி இருந்தா நல்லா இருக்கும் தான் நான் சொல்றேன், இப்படி தான் இருக்கணும்னு நான் சொல்லல, ரைட்? ஸோ ஜஸ்ட் ஹேவ் ஒரு விஷன் மிஷன் வச்சுக்கோங்க, ரைட்? ஸோ கைண்ட் ஆஃப் அ திங் வேர் like just focus on your goals, and you have a lot of opportunities, right? Like we are in an era of like artificial intelligence. எத்தனை பேருக்கு அந்த சாட் ஜிபிடி தெரியும்? எல்லாரும் அதை பத்தி எல்லாம் தெரிஞ்சுக்கிறீங்கல்ல, ரைட்? இப்போ எக்ஸாமே வந்து அதில் எழுதலாம்னு சொல்லிட்டு people have found that like no Siri, no Alexa, right? like it's going to be like ஒரு artificial intelligence இதுல வந்துட்டோம். So try to develop your skills. You need to see, developing a skill is என்ன சொல்றேன், secondary. Primary is you need to update your skill sets. இன்னமும் உட்காந்துட்டு நம்ம வந்து பிசிஆர் நினைச்சிட்டோம்னா அப்படியே தான் இருக்கணும். பிசிஆர் வந்து வெஸ்டர்ன் அண்ட் ஈஸ்டர்ன் கண்ட்ரீஸ்லாம் பார்த்தீங்கன்னா அதெல்லாம் தாண்டி அவன் எங்கேயோ போயிட்டான். நம்ம இப்போதான் வந்து we are talking about PCR, right? like try to develop your skill set and also upgrade your skill set. That is going to be very very important, right? Rather than like sitting on that அதே PCR தான் போட்டுட்டு இருப்பேன் அப்படின்றதுலாம் கிடையவே கிடையாது. Try to develop that. So there are more and more opportunities for microbiologists, and there's going to be huge scope even now. Like last week I was there in some meeting there, in Trivandrum University. There was kind of a talk about space microbiology. புதுசா இருக்குல்ல, space microbiology, right? like now எல்லாருமே வந்து பாத்தீங்கன்னா ஒரு காலத்துல வந்து பாத்தீங்கன்னா it's not a rocket science அப்படின்னுவாங்க. இந்த ராக்கெட் சயின்ஸ் வாஸ் ஒரு டஃபஸ்ட் சப்ஜெக்ட். எனக்கு என்ன பொறுத்த வரைக்கும் டஃபஸ்ட் சப்ஜெக்ட் எதுன்னு பார்த்தீங்கன்னா நியூரோ சயின்ஸ் தான். நான் அதை பத்தி பேச விரும்பல. நியூரோ சயின்ஸ் இஸ் த டஃபஸ்ட் சப்ஜெக்ட், ரைட்? யூ கேன் நாட் ப்ரிடிக்ட் வாட் இஸ் ஹேப்பனிங் இன் யுவர் பிரெயின். யூ கேன் நாட் ப்ரிடிக்ட், என்ன சொல்றது, என்ன நடக்குதுன்னே நம்மளுக்கு தெரியாது, எந்த சிக்னல் தெரியும். ஸோ தட் இஸ் த டஃபஸ்ட். பட் ராக்கெட் சயின்ஸ் ஏன் இப்போ சொல்லக்கூடாதுன்னா, இவன் 10th standard ஸ்டூடெண்ட், ரைட்? லைக் பாத்தீங்கன்னா ராக்கெட், தே டிசைன் த ராக்கெட். ஸோ பார்த்தீங்கன்னா இன்னும் ஒரு 10-20 yearsல we are going to see, right? like space industry is going to boom like anything. பட் அங்கேயும் வந்து பார்த்தீங்கன்னா மைக்ரோபியாலஜிஸ்ட் தேவை, right? So why I'm insisting that like microbiology is important for the space microbiology also, right? I mean space industry இல்லைன்னா, so you would have read about this evolution பார்த்துருப்பீங்க. இந்த உலகத்திலே வந்து பாத்தீங்கன்னா முத வந்து பாத்தீங்கன்னா என்ன ஆர்கானிசம் தெரியுங்களா? What is the organism which is evolved? பாத்தீங்கன்னா ஒன்றும் இல்லை, சைனோபாக்டீரியான்னு கேள்விப்பட்டிருக்கீங்களா? ஸோ யூ ஷுட் ஃபீல் ப்ரௌட் தட் என்ன சொல்றது, மைக்ரோபியாலஜிஸ்ட் தான் முதல்ல, அதுக்கப்புறம் தான் மெடிசன் எல்லாம், புரியுதுங்களா? அதை விட்டுட்டு டோன்ட் ஃபீல் இன்ஃபீரியர். ஏன்னா நிறைய பேர் வந்து பாத்தீங்கன்னா பேரண்ட்ஸோட கம்பல்ஷன்ல வந்து நிறைய பேர் இந்த கோர்ஸ் ஜாயின் பண்ணியிருப்பாங்க, தெரிஞ்சோ தெரியாமலோ வந்து ஜாயின் பண்ணிட்டீங்க, ஆனா நல்ல விஷயம் பண்ணிருக்கீங்க. பட் ஒன்லி திங் இஸ் ரைட், ட்ரை டு டெவலப் யுவர் ஸ்கில் செட்ஸ். Opportunity will come and knock your door. So thank you very much for your kind attention. Thank you.
Thank you, sir, for your valuable words. Now, I request Dr. Ganesh, Scientist E Deputy Director, ICMR, to share us a few words.
Good evening, all. So, first, on the outset, I would like to thank the management as well as the head of the Department of Microbiology, Dr. Vidya, and then the organizing team for giving me this opportunity to come and present whatever research activities we have carried out and some of the activities which I gone through to reach this position. So I just want to tell you the same thing, the opportunity for us will come. If it doesn't come, you try to make an opportunity, but try to use it, and all the best for all your future endeavors. So how many of you are willing to take up research, or if you want to join service in ICMR or many other research institutions in India, like CSIR, ICAR, IVAR, many things are there. I just told about in my presentation. யாராவது தூங்காம கவனிச்சிருக்கீங்க? ஞாபகம் இருக்கா? ஐசிஎம்ஆர்ல எத்தனை இன்ஸ்டியூட் இருக்கு? சயின்டிஸ்ட் பொசிஷன், சயின்டிஸ்ட் பிக்கு எம்எஸ்சி ஃபர்ஸ்ட் கிளாஸ் இருந்தா போதும், என்ட்ரன்ஸ் எக்ஸாம் வச்சு இன்டர்வியூ வச்சு எடுப்பாங்க, சயின்டிஸ்ட் ஆயிடலாம். அதே மாதிரி சிஎஸ்ஐஆர், கவுன்சில் ஃபார் சயின்டிஃபிக் அண்ட் இண்டஸ்ட்ரியல் ரிசர்ச், அதில் எம்எஸ்சி மைக்ரோபயாலஜி, பயோகெமிஸ்ட்ரி, பயோடெக்னாலஜி, இம்யூனாலஜி, மாலிகுலர் பயாலஜி, பயாலஜி, பயோசயின்ஸ், லைஃப் சயின்ஸ், பயோஇன்ஃபர்மேட்டிக்ஸ். இன் அடிஷன் இது இல்லாமல் நான் மெடிக்கலில் சோஷியாலஜி, ஸ்டாட்டிஸ்டிக்ஸ், பயோஸ்டாட்டிஸ்டிக்ஸ் இதெல்லாம் கூட இருக்கு. ஸோ you can use it for yourself or you can tell your friends in this course. அப்போ நீங்களும் சயின்டிஸ்டா ஐசிஎம்ஆர்லையோ இல்லை சிஎஸ்ஐஆர்லையோ இல்லை ஐசிஏஆர், இந்தியன் கவுன்சில் ஆஃப் அக்ரிகல்ச்சர் ரிசர்ச், அந்த மாதிரி நிறைய இன்ஸ்டிடியூட்ஸ் இருக்கு, அதில் யூ கேன் ஜாயின். இல்லாட்டி சிஎஸ்ஐஆர் அண்ட் ஐசிஎம்ஆர் இவங்கெல்லாம் சேர்ந்து நெட் எக்ஸாம், நேஷனல் எலிஜிபிலிட்டி டெஸ்ட் ஃபார் எம்எஸ்சி கேண்டிடேட்ஸ். பயோடெக்னாலஜிக்கு டிபிடி இஸ் கண்டக்டிங் டிபிடி நெட், நேஷனல் எலிஜிபிலிட்டி டெஸ்ட். அதில் 5 years ஃபெலோஷிப். ஃபர்ஸ்ட் 2 yearsக்கு ஜூனியர் ரிசர்ச் ஃபெலோ, அப்புறம் 3rd yearலேருந்து 5th year வரைக்கும் சீனியர் ரிசர்ச் ஃபெலோ. யூ வில் கெட் அ குட் அமௌண்ட் ஆஃப் ஸ்டைபண்ட் அலாங் வித் ரிசர்ச் ஃபண்ட்ஸ். நீங்கள் ஏதாவது இன்ஸ்டியூட்டில் போய் ஜாயின் பண்ணி 5 yearsல பிஹெச்டியை முடிச்சிடலாம். ரைட்? இன்ட்ரெஸ்ட் இருக்கா? ரிசர்ச்சில் யாருக்கா இன்ட்ரெஸ்ட் இருக்கா? டைரெக்டாக சயின்டிஸ்ட் ஆனாலும் ஆகலாம், பிஹெச்டி பண்ணிட்டும் ஆகலாம். ஃபர்ஸ்ட் கிளாஸ் எம்எஸ்சி டிகிரி இருந்தாவே யூ ஆர் எலிஜிபிள் டு அப்ளை ஃபார் சயின்டிஸ்ட் பி பொசிஷன், which is non-medical, belong to microbiology, biochemistry and the category. Second class MSc plus PhD இருந்தாலும் same post you are eligible to apply. Join பண்ணிட்டும் PhD பண்ணலாம், PhD பண்ணிட்டும் join பண்ணலாம். புரியுதா? இன்ட்ரெஸ்டடா? ஓகே, ஆல் தி பெஸ்ட். இந்த மாதிரி ஒரு நல்ல ஆப்பர்ச்சுனிட்டி வந்திருக்கு. நாட் ஓன்லி டிஜி வைஷ்ணவ காலேஜ்ல இல்லாம வேற அதர் காலேஜஸ்ல இருந்தும் நிறைய கேண்டிடேட்ஸ் வந்திருக்காங்க. எந்துவா ப்ரெசென்ட் பண்ணிருக்கீங்க. நேற்று என்னால் வர முடியல, நேற்று வேற ஒர்க்ல இருந்து மேடம் கூப்பிட்டுருந்தாங்க, வந்தா ஜாயின் பண்ணலாம் அப்படின்னாங்க. அதே மாதிரி இன்டராக்ஷன், அதே மாதிரி இன்டர்ன்ஷிப் இருக்கு. எம்எஸ்சி ஃபைனல் இயர்ல வி ஹேவ் 3 months இன்டர்ன்ஷிப் இருக்கு ஃபைனல் இயர் தீசிஸ் டிசர்டேஷனுக்கு. இஃப் தே ஆர் பிடெக் பயோடெக்கா இருந்தா அவங்களுக்கு 5 months வரைக்கும் இருக்கு, 3 to 6 months வரைக்கும் இருக்கு இன்டர்ன்ஷிப் பண்ண. ஒரு சென்டர், ஒரு காலேஜ்ல இருந்து 2 பேர் இல்ல 4 பேர் தான் எடுப்பாங்க. வி ஹேவ் 3 faculties தான் லேப்ல, என்ஐஏ சொல்றேன். அதே மாதிரி ஐசிஎம்ஆர் இன்ஸ்டியூட்ல சென்னையில சேத்பட்ல ஒன்று இருக்கு, டியூபர்குலோசிஸ் ரிசர்ச் சென்டர். அங்கேயும் நீங்க இன்டர்ன்ஷிப் பாக்டீரியாலஜி, வைராலஜி நிறைய டிவிஷன்ஸ் இருக்கு. ஸோ யூ கேன் ஆல்சோ அப்ளை ஃபார் ஃபைனல் இயர் தீசிஸ் டிசர்டேஷன் இருக்கு இல்லையா, அதுக்கு யூ கேன் சப்மிட். மோரோவர் இன்னும் ஒன்று சொன்னார் சார், நீங்க எந்த இன்ஸ்டியூட்ல பண்றீங்க, பிரீமியர் ரிசர்ச் இன்ஸ்டியூட்ல தான் போய் பண்ணணும்னு அவசியம் இல்லை. பட் வாட் எவர் திங்ஸ் யூ டூ, அந்த ப்ராப்பரா சிஸ்டமேட்டிக்கா நீங்க பண்ற டிசர்டேஷன் வில் பி யுவர் ஓன் எக்ஸ்பீரியன்ஸ். ரிசர்ச்சுக்கோ நெக்ஸ்ட் ஃபியூச்சர் ஜாபுக்கோ கூட ரொம்ப யூஸ்ஃபுல்லாக இருக்கும். ஸோ நம்ம பண்ணுற வேலையை தெளிவாக புரிஞ்சுட்டு எந்த ஸ்டெப் எதுக்கு பண்ணுறாங்க ப்ராப்பராக சிஸ்டமேட்டிக்காக பண்ணிங்கன்னா லைஃப்பில் எப்பயுமே யூஸ்ஃபுல்லாக இருக்கும். படிக்கிறது எஜுகேஷன் வந்து வயசு இல்லை, படிக்கிறதுக்கு வயசு இல்லை, நாலேஜ் கெயின் பண்ணுறதுக்கும் வயசு ஒரு லிமிட்டே கிடையாது. ஜாப் ஆப்பர்ச்சுனிட்டி கேட்டாங்க 35 இயர்ஸ்க்கு மேலே அப்படி இப்படின்னா, அதெல்லாம் ஒன்றும் இல்லை. யூஆர் எலிஜிபிள் டு அப்ளை, கோர்ஸ் கம்ப்ளீட் பண்ணிங்கன்னா எலிஜிபிள் பண்ணலாம், யூ கேன் ஜாயின் த போஸ்ட். ஸோ மேலே அதே மாதிரி ரிசர்ச்சுக்கும் ஒரு இன்ட்ரெஸ்ட் இருக்கணும், டுவர்ட்ஸ் ஒரு எய்ம் ஃபிக்ஸ் பண்ணிட்டு அப்துல் கலாம் என்ன சொல்லியிருக்காரு, நீங்க கனவு காணுங்கள், கனவு வந்து தூக்கத்துல வரும்போது வர்ற கனவு இல்லை, உங்களை தூங்க விடாம செய்வது எதுவோ அதுதான் கனவுன்னு சொல்லியிருக்காரு. ஸோ யூ ஆல் ஷுட் ஹேவ் ஒன் ப்ராப்பர் கோல், ஒரு டார்கெட் அச்சீவ் பண்ணி டார்கெட் ஃபிக்ஸ் பண்ணி அதை நோக்கி பயணிச்சிங்கன்னா உங்க லைஃப் ரொம்ப சக்சஸ்ஃபுல்லா போகும். ஸ்கை இஸ் அ லிமிட், வானம் தான் எல்லை. எல்லாரும் எந்த ஃபீல்டில் வேணா போகலாம், பட் நீங்க எடுக்கிற ஃபீல்டு நல்ல ஃபீல்டு, அதுலேயும் நீங்க ரிசர்ச்சும் பண்ணலாம், இல்லை டீச்சிங்கா அகாடமிக்கா என்னன்னு பார்த்து டிசைட் பண்ணி பண்ணுங்க. யூஸ் பண்ணுங்க உங்களோட சீட்டை. தேங்க்யூ ஆல்.
Thank you, sir, for your valuable speech. It's time to announce the prize winners of poster and paper presentation. Now, I kindly request Dr. Prabhu, Head of the Department, University of Madras, to distribute the prize winners of paper and poster presentation. For paper presentation, second prize is shared by both of them. Jeevita M. Quaidi Millet, Government College for Women, Guest Scholar, Department of Plant Biology and Plant Biotechnology. And Satyasini, Research Scholar, Department of Chemistry, Dwarkadas Govardhandas, Vaishnav College. And the first prize goes to Priyamvada, Vellore Institute of Technology, Department of Bioscience. For poster presentation, the second prize goes to Lakshmi Priya S, PG and Research Department of Microbiology, Dwarkadas Govardhandas, Vaishnav College. And the first prize goes to Idel Queen J, Research Scholar, Department of Chemistry, Dwarkadas Govardhandas, Vaishnav College. Thank you.
I kindly request our dear participants to come and share your view regarding the conference. Anyone interested to share?
யாராவது இன்ட்ரெஸ்டடா இருக்கீங்களா?
From Apollo, who can speak?
Good evening to one and all present here. I am Shankita from Apollo Arts and Science College, Chennai. First of all, I thank DG Vaishnava College for this wonderful opportunity. This international conference is able to help to develop our skills through the microbiology department. And I thank all the scholars and technical speakers to engage some interesting facts about the microbiology, and thanks for the wonderful opportunity.
to all of you. So first of all, I would like to congratulate the Department of Microbiology, Deji Vaishnav College on the 25th anniversary and for conducting this very auspicious event. Conferences like this are a boiling pot of ideas. So it was very interesting to know the various viewpoints of the presenters, the resource persons, and all those who presented their papers and posters. I would like to congratulate you all as well for coming up and presenting the papers. And obviously, our chief guests and our resource persons, I'm very grateful to all of you. You have come here to enlighten us about the vast field of microbiology that it is. And I'm sure that we as students will take a note of this and use it to develop and to get more opportunities for us in the future. So thank you one and all once again for this opportunity.
சீக்கிரம் போ. ஸ்லோ மோஷன்ல போயிட்டு இருக்கேன். நானும் பாக்குறேன் போவா போ.
May I now call upon Dr. Raj Lakshmi, Assistant Professor, Department of Microbiology, to deliver the vote of thanks.
Good evening, everyone. Feeling gratitude and not expressing it is like wrapping a present and not giving it. I am going to express my heartfelt gratitude. As we come to the closure of the International Conference on Next Generation Microbiologist Challenges and Opportunities 2.0, I deem it a great honor to propose the vote of thanks to all who have helped in making this event such a resounding success. On behalf of the organizing committee, I convey my deep regards and hearty thanks to our secretary, Dr. Ashok Kumar Mundraji. Words are not enough to thank his constant guidance and support to shape the conference. My sincere thanks to our principal, Dr. S. Santosh Babu, who have helped us obtain the necessary administrative approval for organizing such an event in our campus. He supported us in all possible ways to organize this conference. Thank you, sir. I would like to thank all the guest speakers and delegates who shared their knowledge and their valuable time with us. I feel deeply gratified and thank Dr. Prabhu, Chief Guest, for the valedictory function, for accepting our invitation and for gracing this occasion. Thank you, sir. I would like to thank our beloved Head of Department, Dr. P. Vidya, for her constant support and the guidance that radiated a source of energy within us. We will ever remain grateful to you, ma'am. I am very much thankful to all our faculties and non-teaching staff members for making this event a successful one. An event of this dimension cannot happen overnight. The weeks started rolling months in advance. We have been fortunate enough to be backed by a team of dedicated students committee and research scholars. I cannot thank everyone enough for the involvement they have shown and the willingness they have expressed to take on the completion of tasks beyond their comfort zones. Thank you all. At the end, I would like to take the opportunity to thank each and everyone for their support and make this event a successful one. I would like to thank all the participants who have participated overwhelmingly and helped them in making the event a successful one. If time is money, then today you have spent millions for us. Thanks to all of you for making this event a grand success. Once again, I thank you all for the cordial cooperation. Thank you.
Now, I request you all to stand up in respect to our national anthem.
तव शुभ नामे गाहे, तव शुभ आशीष माहे।